Hello students, welcome back to the channel. In this video, we are going to study about stamen, which is a part 2 of the sexual reproduction in flowering plant. Now see, male reproductive organ of flowering plant is androecium. And its unit is known as the stamen, which is also known as microsporophyll. And each stamen typically will have two parts. One is filament and anther lobes. And in between that, connective is present. And connective contains only vascular tissue, means xylem and phloem, and it is sterile. So in our NCERT, this is the diagram of connective. So you should know it is present between the filament and anther lobe, and sometimes present between the two anther lobe, also extending up till there. And it is a sterile tissue. Clear? Now filament is actually long stalk, and it is joining the anther lobes to the thalamus. This is thalamus. Clear? So it is attaching the anther lobes to the thalamus, and this part of the filament is known as proximal end. Please understand, this was PMT question. The proximal end of filament is attached to, obviously answer is diagram is helping you to learn, it is thalamus. So proximal end of filament attached to thalamus, and it can attach to petal also in the family Solanaceae. Okay, in the family Solanaceae, you can check your morphology 11th class, you will find it is attached to petal. Then if you see the filaments, they are showing variation in the number also. They show variation in number. For example, family Fabaceae is having 10 stamen, Solanaceae 5, Liliaceae 6. So number are variable, and even the length of stamen is also variable. And we have studied length of the stamen is variable because of the difference in the length of the filament in two examples in the morphology chapter, that is salvia and mustard. So they are showing variation in the number, they are showing variation in the length, and obviously the way of attachment also, size and shape. Now, which is the fertile part of the stamen? Fertile part of the stamen is anther lobe. Fertile part is anther, which is the knob-like structure, and in typically angiosperm, you will find it is having two lobes. So it is known as bilobed structure. And each lobe has two theca. Each lobe has two theca. Please understand, it's very important question. Each lobe has two theca. We say it is dithecous. Clear? You can understand this way. These are the two anther lobes. Okay. And in each anther lobes, you are finding this is a two pollen sac, means microsporangia. And this is the filament. So who is separating the two theca? Longitudinal groove. Again, PMT question. Which groove separate? It's a longitudinal groove which separate the two theca. And each lobe has two theca. Please understand, each lobe has two theca. And if you want to study the bilobed nature, you will see more clearly in the transverse section, not in the longitudinal section. Again, important question. Now, anther, if you see, it is four-sided organ. So come back to the diagram. Please see the diagram carefully. You will find it is four corners. Check. It is four-sided structure. We say it is tetragonal structure, and at each lobe, it is two pollen sacs are present. These are pollen sac, means microsporangia are present. So two in each lobe. So anther is four-sided, means tetragonal, consisting of total four microsporangia, and two in each lobe. We will say it is tetrasporangiate. Total four sporangia, we will say tetrasporangiate. So what are the points you have to remember? Okay, first of all, anther is dithecous, and it is tetrasporangiate. Clear? Very important question. Please learn it. Microsporangia will develop and become pollen sac, and pollen sac will appear circular in outline in transverse section. In transverse section, they will appear circular in outline. Now, again, very important concept. See, we are saying here, anther is analogous to sorus of fern. So first of all, you should know what is sorus. Collection of sporangia, collection of sporangia is known as sorus. You have recall in the plant kingdom we studied sporophyll has sporangia. And if there are many sporangia, we will say sorus. So here right now, anther is having 4 sporangia. So we can say it is analogous to sorus of fern. This is a general. But some exceptions are there. You will see monothecus. If I am saying monothecus means it is having only one anther lobe. Is it clear? It is one anther lobe. And it is bisporangiate, seen in family Malvaceae, which is again your neat family's questions. Members are China rose, ladyfinger, cotton. Monothecus and monosporangiate condition is seen in Arcithobium. Now, at maturity, you will find that the sterile tissue which was separating between the two pollen sac, they degenerate. So you can see here in the diagram, you will find only one locule here and one locule is here. So this is a mature dehiscent anther. So at the time of the mature dehiscent, you will find it is dithecous, it is tetrasporangiate, but it is not tetralocular. You will see it is bilocular. Concept is getting clear because the sterile tissue which was present between the two pollen sac of one anther lobe is now degenerated and you will find only one locule in each anther lobe. So bilocular. Now, development of anther is eusporangiate. Eusporangiate means it is made from the group of the cell. So you will find, see here in the diagram, it is generally appearing circular. You will find this first layer is known as epidermal layer. This is epidermal layer. And below this, you will find in the hypodermis region, you are finding at 4 corner archosporial cells are differentiated. Okay, these archosporial cells are differentiated and NCERT has used this term in the summary with respect to the female part. So you can check in the summary, it is used by the NCERT. Now, archosporial cell, let me explain you. This is one archosporial cell. It will go for periclinical division. Means it will divide in the circle to make 2 cells. Outer parietal cell. And this is inner sporogenous cell. Outer parietal cell will give rise to wall layer. It will give rise to wall layer by many periclinical division and anticlinal division. Means it will go for many periclinical division. Let me make this way periclinical division. Okay, and anticlinal means on the side this way. Let us assume this is your sporogenous cell present. Center is a sporogenous cell present. It has not become into tissue, but later on it will also become into the sporogenous tissue because a sporogenous cell will make a sporogenous tissue. So what are the anther walls? If you see, first is epidermis, second is endothecium, third is middle layer. Okay, middle layer, 2 arrows indicate it is more than 1 layer because epidermis only 1 layer, endothecium only 1 layer, but middle layer is 2 to 3 layers. And innermost is tapetum. And this is a sporogenous tissue which is made from sporogenous cell by many periclinal as well as anticlinal division. Is it clear? So I hope so this much is comfortable for you. Let me explain you. Let us check the concept, how much concept we have understood. See, these are the questions. In a transverse section, a typical microsporangia appear dash in outline. Is it circular, tetragonal, oblate, or rectangular? If you check right now, we discuss it will be circular. Now, which end of filament is attached to the thalamus or petal? I focus it's a PMT question. Answer will be proximal end. Next, theca is separated by which groove? I focus it is, if you remember in the theory while explaining you, it's a longitudinal groove which separate the two theca. Now, all of the given statement are correct about stamen, but one is not correct. I think so. First is correct. Check. Bilobed nature is not seen in the longitudinal section. Let me show you back once again. Check bilobed nature. Let me highlight it with different color. See this color. Bilobed nature is more clearly seen in transverse section. Is it clear? It is more clearly seen in transverse section. So it is not longitudinal, it is transverse section. So this statement is only the one which is incorrect, because they're saying all are correct except. So you have to find which is incorrect. Clear? So answer will be the second option. Number, this is also correct statement. Anther is tetragonal, correct, having 4 microsporangia, 2 in each lobe. Yes, this is also correct. So answer question was asking which is incorrect. So I hope so you are able to get the answer also. This video might be helpful for you. Thanks for watching. In the next video, we are going to discuss about the anther wall more in detail.